Welcome, everyone. Today's mini talk is a foundations overview of Mycoplasma, a group of bacteria in the molecules that matters a great deal in veterinary medicine. The single most important thing to remember is that these organisms have no cell wall, which shapes everything from how they look under the microscope to how we treat them. They live a minimalist lifestyle on the mucosal surfaces of their animal hosts and cause chronic, hard to clear infections. Over the next few slides, we'll cover their unusual biology, the key species across cattle, poultry, swine, and pets, how infections present and spread, and how we diagnose, treat, and control them. I'll keep the emphasis on the core principles you'll carry into clinical work. Let's get to it. It explains what makes mycoplasmas biologically unique. They are among the smallest self-replicating bacteria known, bounded only by a sterile containing membrane rather than a rigid cell wall, which is why they are pleomorphic and stain poorly with Gram stain. Their tiny genome leaves them with limited metabolism and many dependencies on the host, so they specialize in adhering to mucosal surfaces such as the respiratory and urogenital tracts, the mammary gland, joints, and even red blood cells in the hemoplasmas. Through intracellular survival, antigenic variation, and immune modulation, they establish chronic, persistent infections that are hard to clear. The clinical punchline is critical. Because there's no cell wall, penicillins, cephalosporins, and other beta-lactams simply do not work. They target a structure Mycoplasma doesn't have. We walk through the major veterinary hosts, each with its own signature species. In cattle, M. bovis drives pneumonia, mastitis, and arthritis in dairy and beef herds. Chronic, costly, and slow to respond to treatment. In poultry, M. gallisepticum causes chronic respiratory disease in chickens and infectious sinusitis in turkeys, lowering both egg and meat output. In swine, M. hypopneumoniae produces enzootic pneumonia marked by a dry cough and reduced growth, and opens the door to the broader porcine respiratory disease complex. Cats face M. haemophilus, a hemotropic species that coats red blood cells and causes feline infectious anemia. It's worth flagging a few other notable species too: M. synoviae in poultry, M. agalactiae in sheep and goats, and M. mycoides, the agent of contagious bovine pleuropneumonia. This slide covers how mycoplasma infections actually present in the field and how they move through a herd or flock. On the clinical side, watch for chronic respiratory disease with coughing, nasal discharge, and poor weight gain, mastitis and a sharp drop in milk yield in M. bovis dairy herds, swollen painful joints and lameness from polyarthritis, and hemolytic anemia, weakness and fever in hemoplasma infections. A key point is that infection is often subclinical, so silent carriers can quietly seed an entire group. Transmission happens through direct contact and inhaled aerosols, vertical or in ovo spread, colostrum and milk, contaminated equipment and fomites, and especially the introduction of carrier animals into a clean group. Remember that crowding, stress, and co-infections all accelerate outbreaks, which is why management is as important as the pathogen itself. We turn to the diagnostic workup and treatment. For detection, PCR is the fast, sensitive frontline test for finding and typing Mycoplasma DNA. Culture is definitive but slow and fastidious, needing special media and days to weeks to grow. And serology with ELISA or HI is used to screen herds and flocks for exposure and surveillance. On the treatment side, the guiding principle is to choose antibiotics that bypass the missing cell wall. Macrolides, tetracyclines, fluoroquinolones, and pleuromutilins all act on protein synthesis or DNA. Beta-lactams, like penicillin and cephalosporins, are useless here because they target a wall these bacteria don't have. Finally, resistance is rising, and that treatment usually reduces clinical signs, but rarely fully clears the infection. So it must be paired with good management and control. And the key takeaways with the theme that prevention beats cure. Biosecurity is the foundation. Quarantine and test new arrivals and control the movement of animals and equipment. All in, all out batch management with cleaning between groups breaks the cycle of spread, and vaccination is available for some species, such as M. gallisepticum in layers and M. hypopneumoniae in pigs. Routine PCR and serology keep herds and flocks under surveillance and disease-free. So 3 core messages. No cell wall means no beta-lactams, so therapy relies on macrolides, tetracyclines, and fluoroquinolones. One genus reaches many hosts, with cattle, poultry, swine, and pets, each facing their own species, and because infection is chronic and costly, biosecurity and surveillance are where the real wins lie. If you found this information helpful, like and subscribe. Thanks, and I'll see you next time.